Hello students, welcome to Distinction Learning Platform, and on this platform you'll find revisions, you'll find explanation to difficult topics, you'll find tests, quizzes, a lot of stuff. So before we begin our lessons, make sure that you subscribe so that you can be updated with our upcoming videos. To make the story short, let us begin. So in this video, we're talking about irreversible cell injury, and one of the type of irreversible cell injury is apoptosis. Remember that we talked about necrosis and all the types of necrosis, or pattern necrosis, morphological features as well. Okay. So in this video, we're talking about apoptosis. What is apoptosis? Okay. Apoptosis, basically, we said this is the programmed cell death. So it's a type of it's a way in which a cell can die which is already programmed. Okay. So what are we saying? What is apoptosis? So this is what apoptosis is: programmed cell death. Okay. What is apoptosis? Apoptosis is what? Programmed cell death. So now, what are the causes of this programmed cell death? There are a lot of causes of programmed cell death, but they are categorized into two categories. The first one is what? Physiological causes, and the second one is what? Pathological causes. And what do we mean if you say physiological causes? If you say physiological causes, basically these are the normal causes of apoptosis. Basically, things that we consider normal. Pathological causes are what? It is due to what? A certain underlying condition or a disease that is causing a cell to die on its own, or it's being programmed so that it can die. Okay, so that's under pathological causes. So we're going to begin with what we call physiological causes. So what are the physiological causes? Okay, what is the first one? The first one is remember, during the embryogenesis or developmental process, there is what? There are some cells that are going to disappear. Basically, what we mean is they are going to die. So there are what we call there are web tissues between the fingers and the toes. They are going to disappear by the cell death called what? Apoptosis. So what are we saying? What's the first physiological cause? The first physiological cause is what? Embryogenesis. During embryogenesis, there is what? Disappearing of what? There is what? Disappearing of web tissues. Okay? Disappearing of what? Web tissues between the what? The fingers and the what? And the toes are going to disappear. A lot of stuffs or tissues are going to disappear during what? Embryogenesis. Basically, that is normal. Okay? What else apart from embryogenesis? So what if if you look at a good example, if you look at during menstrual cycle or during the menstruation period in females, there is what? Endometrial shedding. This endometrial cells, they are going to die by apoptosis, and they are going to shed off during the menstrual period. Okay, so that is also the normal thing because it happens in females. Okay, so what are we saying? So basically what we are saying is just the withdrawal of stimuli. So if you remove a stimuli from a cell basically is going to die. And if you are talking about that good example under menstrual cycle, so if you withdraw the stimuli, like if you withdraw the progesterone hormone, if you withdraw the estrogen hormone, then the cells in the endometrial cavity there, they're going to what? They're going to start shedding off. They are going to die by apoptosis. Okay. So apart from the withdrawal of that stimuli under that condition, also there is at the end of the acute inflammatory response or the chronic inflammatory response, all the cells, all the white blood cells that were activated, they are going to what? To die by apoptosis as well. So that is also the withdrawal of what? The stimuli. Okay, two good examples. Okay, what else apart from that? Apart from the withdrawal of the stimuli, embryogenesis, what else? There's also another physiological cause of apoptosis where where they're under immune system, they are what we call self-reactive what leukocyte or what blood cells. They are going to die because they are able to recognize what the self antigens. Okay, if they recognize the self antigen, basically they start eating up those cells. Okay, or phagocytose those cells. So to prevent that, the immune system undergoes what we call selectivity. Okay, so they try to eliminate those what harmful self-reactive what leukocytes. So what are we saying? Also, what we're saying is removal of self-reactive white blood cells or leukocytes. Okay, basically those T cells, B cells, they're going to be what? Removed as well by that process. Okay, so basically 3 causes of what? Apoptosis, physiological causes of apoptosis. Okay, so now let's talk about now the very, very important part, the pathological causes of what? Apoptosis. Pathological causes of apoptosis. So what is a pathological cause of apoptosis? Basically it is because of what? An underlying disorder. Okay, so what is the first thing, what is the first pathological cause of apoptosis? Basically we know, you know, if the DNA is severe damage because of maybe a lot of causes, maybe anti-cancer drugs, it was exposed to radiation, it was exposed to free radicals, then the DNA is going to be what? Damaged. So what are we saying? Severe damage to what? DNA. The first one is what? Severe damage to DNA. Severe damage to DNA. Okay, that's the first cause. So the first cause is what? Severe damage to DNA. What can cause severe damage to DNA? We are saying if a cell is exposed to severe hypoxia, or it was exposed to what? Radiation. Maybe it is exposed to what? Maybe some anti-cancer drugs, and maybe a lot of other environmental factors. So those can cause what? DNA damage. If remember that if DNA is damaged, basically beyond the repair, then what's going to happen? Because there is a certain suppressor gene that is there. So there are some suppressor gene that is there, which is called TP53. So TP53 is the one that decides if a DNA is damaged. It's the one that triggers DNA repair. So it will tell the DNA to say, okay, let's try to repair this damaged DNA. If we fail to repair the DNA which is damaged, then I'm going to induce what we call apoptosis. So basically what I'm saying is that there's the gene that is there that's going to what? To decide whether a cell will undergo apoptosis or will undergo DNA repair. Basically, if it fails to undergo DNA repair, it is going to initiate what we call apoptosis. Okay, that's what we call TP53 gene, and it's a very, very important gene because there are some cancers that are going to mutate that TP53, because if TP53 is become mutated or undergoes mutation, then what's going to happen? Then it will cause what? Cancer. And those are the other mechanisms that are going to cause what? Cancer. So we'll talk about cancer in future. Okay, so that's under what? Severe damage due to DNA, leading to what? Apoptosis. Remember that severe damage to DNA, that should be under a pathological cause. Okay, damaging to what? DNA. What else? So what else apart from severe damage to DNA? Okay, there's another one. Okay, accumulation of what? Misfolded proteins. Okay, accumulation of what? Misfolded proteins in the body. Okay, so accumulation of misfolded protein, it can be caused by a lot of causes. Maybe some genes there that are defective, genes that are making proteins and they are defective, then it can lead to what? Accumulation of misfolded protein, as well as also some extrinsic factors like what? Maybe the reactive oxygen species, they can also cause what? Misfolded protein, and as well as what? The hypoxia can also cause what? Misfolded proteins. So basically what we're saying is that misfolded proteins can also be a pathological cause. And remember that misfolded protein, what are the three diseases that you can see? The diseases that can show misfolded proteins. The first one is what? Parkinson's disease. The second one is what? Huntington what? Disease. Okay. And the last one is what? Alzheimer's disease. So these are three important disorders that shows what? Misfolded proteins. And basically, if a cell is shown misfolded protein, basically it's going to activate what? It's going to activate the apoptosis process, causing a cell to die by suicide. Okay, very, very important what? Point. Okay, so that's under misfolded proteins. What else? What is the other cause? The other cause is this one. Okay, the other cause is this one. What if a cell is viral infected? So also infection can cause what? Apoptosis. If a cell is viral infected, so it dies itself intrinsically, inducing like intrinsic type of apoptosis. Okay, then either maybe the white blood cells, they will come and kill the viral infected cell. Okay, so what are we saying? Also, viral infected cell can cause what? Apoptosis. Either what? We said that virus, like the HIV virus, that DNA of the HIV virus can embed itself into the DNA of the host DNA. Then the host DNA can get damaged, then causing what? Initiating what? Apoptosis. Okay, what else? Then also this viral infected cell, from the cells, the white blood cells or the leukocytes that are nearby, they can recognize that viral infected cell and kill it by what? Inducing apoptosis via the what? The extrinsic pathway of what? Apoptosis. We will talk about that. Okay. So basically those are the what? Those are the pathological causes of what? Apoptosis. Although there are a lot of pathological causes of what? Apoptosis. But basically these are what? Very, very important what? Pathological causes. Okay. So now, what else? So now let's talk about the mechanism of apoptosis, and this is the very, very, very important what? Part. Okay. Make sure that you pay attention here. So now, how does apoptosis happen now? How does apoptosis happen? Okay. So that is now the mechanism. So what is that the mechanism of apoptosis? There are two mechanisms of apoptosis. Okay. There is extrinsic pathway and the intrinsic pathway. So what are we saying? So there is what? How many pathways do we know? So there is what? Intrinsic and what? Extrinsic pathway of apoptosis. Okay, so there are two types or two pathways in which apoptosis can happen, either intrinsic pathway and the extrinsic pathway. So we'll talk about both. We'll start first with what? Intrinsic pathway, then ending with what? Extrinsic pathway. Very, very important. Okay. So now, intrinsic pathway, let's start with intrinsic pathway. So now remember that these pathways, they are further categorized into what? Two main parts. Okay. So these pathways are further what? Categorized into what? Two main pathways. What is that? Okay. So this pathway, whether extrinsic or intrinsic, so these pathways, okay, they are further categorized into two. What is the first one? So is what? Initiation pathway, and the second one is what? Execution pathway. So what are we saying? So we are saying the apoptosis pathway is further categorized into two: the initiation pathway and the what? executional pathway. Okay, and there are some certain enzymes that are going to be what activated, because a cell to die on its own, the cell is going to start programming its own enzyme to start eating up the cell and die by what? Apoptosis, like that. Okay, so basically you need to know all those enzymes. So those enzymes are called what? Caspases. Okay, so let's start with this side. So from this side, this is what we call mitochondria. Okay, this part, so this is the mitochondria. Okay, so this whole thing is what we call mitochondria, because it's a very, very important what? Important organelle that is involved in what? Apoptosis. Okay, so let's start with this one. So mitochondria, okay, mitochondria has the inner membrane and the outer membrane. Okay, in the outer membrane, so let's say this part, okay, this part is the outer membrane. Outer membrane. So this whole part here is the outer membrane. Okay, so this outer membrane, there are very, very important proteins that are involved in what we call apoptosis. Make sure that you know, in the outer membrane of the mitochondria, there are some proteins that are very, very important in what? Apoptosis. So I'll write them. Okay, so in this outer membrane, what can you find? Okay, so in this outer membrane, what can you find? There are, so in the outer membrane, so outer membrane, in the outer membrane, what can you find? So they are what we call BCL2 family. So in the outer membrane, they are what we call a BCL2 what family. So that family of proteins, there are about 20 proteins that are there. Okay, and this BCL2 family is the one that controls apoptosis. Okay, either to initiate apoptosis or maybe to inhibit apoptosis. So what are those? So BCL2 family, they are further categorized into two. Okay, into two what? Important family. The first one is what? Pro-apoptotic factors. So the first one is what? Pro-apoptotic proteins or factors. Okay, I'll talk about the examples. And what else? Anti-apoptotic what? Anti-apoptotic factors. Okay. Okay. Whoa. So pro-apoptotic and apoptotic. So let's start with pro-apoptotic. So pro-apoptotic, what does it mean? So pro-apoptotic factors or proteins, these are the proteins that promote apoptosis. So they like inducing apoptosis. And apoptotic factors, these are the proteins or factors that are going to inhibit apoptosis. Basically, they don't like initiating or starting what? Apoptosis. Okay, so now the question is, what are those pro-apoptotic factors? So those pro-apoptotic factors, basically we just talk about two. Okay, the first one is what? BAC. Okay, and the second one is what? BAX. Okay, so what is the other one? And apoptotic factor. So these are apoptotic factors where we just talk about what? Two. Okay, we talk about which is it? The BCL2. Okay, so this BCL2 is not this one. This is BCL2 family, and this is BCL2 only. Okay, what is the other one? BCLXL, like that. Okay, these are the very, very important what factors. Although there are others also, like MCL1, there are other what? And apoptotic factors as well, but these are the main ones. Okay, but there are other proteins that are what also included in under BCL2 family. Very, very important. And these proteins, they control what? These two factors. So these proteins, they control what? These factors. So what do we call them? Okay, so what I'm saying is there is also another group that's going to control, either to promote the pro-apoptotic factors or to promote the what, the anti-apoptotic factors. Okay, so and these proteins are called what? These are called, they are called apoptotic control proteins. They are called a lot of things. Okay, so but what are those good examples of that? So good examples of factors that are going to control pro-apoptotic and the anti-apoptotic factors. Good example are what? Make sure that you know them. BIM, okay, BID, and BAD. So that's very simple. What? Wait, BIM, BID, BAD. Very simple. So these proteins, these are also proteins. So they just control either to promote apoptosis or to inhibit apoptosis. So BAD, BIM, BID. Okay, they control those. Okay, so since we've understood what is there in the outer membrane, so remember that in the outer membrane of the mitochondria, there are what? They are what we call BCL2 family. And remember that BCL2 families can it's not always like in the outer membrane. It can also be found somewhere else, like in the ER. You can find it in the maybe cytoplasmic membrane, there nuclear membrane. You can find them as well. Okay, but the major part where you can find them is in the what? Outer membrane of the what? Mitochondria. Okay, now, so since we've understood something like that, now let's talk about what happens. Because if you talk about what happens from the mitochondria aspect, then we are talking about the intrinsic pathway of what? Apoptosis. That's why sometimes it is called what? The mitochondrial pathway. Okay, so let's start from here. So what happens is like this. Okay, so due to those causes that we talked about, those causes of either physiological or maybe pathological. Okay, maybe there is severe DNA damage, maybe there is maybe viral infected cell, there is severe hypoxia, there is maybe a lot of causes. Okay, if there are those causes, what are they going to do? Okay, so those causes now, what they are going to do is they will start activating what? These control signals. If these control signals now they are activated, then they are going to activate what? The pro-apoptotic what? Factors like what? BAC and the BACs. So if now these BAC and BACs they are activated, okay, in the outer membrane of the what? Of the mitochondria, what are they going to do? They will come here. Okay, so they come here in the on the outer membrane of the what? Mitochondria, and what are they going to do? They will start what? Forming what? Pores. Okay, they'll start forming what? Pores here. Okay, and you just know that this is what? BACs, and this is what? BAC. Okay, so this is what they do because of those same causes that we talked about before apoptosis. If that happens, what's going to happen? BACs and BAC, they're going to be activated by what? BAD, BIM, and what? BID, because these are the one that controls either the cell goes into what? Apoptosis or not going to apoptosis. Now, a cell want to go into apoptosis. What are they going to do? They're going to activate BAC and BACs. If BACs and BAC they are activated, what do? They will come here in the mitochondria and it forms pores in the what? Mitochondria, so that the permeability of the mitochondria becomes it is going to what? Increase. Now, if they form these pores, what's going to happen? Something is going to leak out from the what? From the mitochondria here. Something is going to leak out from the mitochondria. Okay, something is going to leak out like that. Okay, what is that very important thing that's going to leak out from the mitochondria? We call it what? Cytochrome what? C. Okay, so very important point. Okay, cytochrome what? Cytochrome C. And other proteins that are going to what? Promote what? Apoptosis, because there are a lot of proteins in the mitochondria, but the most important thing that's going to leak out after the mitochondria membrane is increased, then is what? Cytochrome C. Make sure that you know cytochrome C is the one that's going to what? To leak out from the mitochondria. Then if this cytochrome C leak out is going to combine with something very important. Okay, it's going to combine with something, a protein that is always found in the what? cytoplasmocytosis of the cell. So what is that? We call APAF. Okay, so APAF. So APAF1, what is APAF1? APAF1 is what? Apoptosis activating factor 1. So basically, let's just call it APAF1. Okay, so APAF1, what's it going to happen? So what's going to happen to APAF1? So APAF1, which is already in the cytoplasm, will start combining with what? Cytochrome C. If it's combined with cytochrome C, okay, it's going to form an important substance. So APAF1 is going to combine with what? Cytochrome C to form what we call apoptosome. Apoptosome. Okay? So if that substance is now produced because it's combined with what? Cytochrome C, then this apoptosome is going to do a very, very important thing. It's going to activate a certain enzyme called what? A caspase, which is caspase 9. There are a lot of caspases. Remember that if you look at these caspases, these are what? Enzymes. So these are enzymes that will help to what? Activate apoptosis. Okay. So these caspases, they are further categorized into what? Two. They are initiator caspases and what? Executioner caspases. Under initiator caspases, I'll just pick two good examples. There's caspase 9 and caspase 8. These are what? Initiator caspases. Executioner caspases, these are what? There is what? Caspase 3 and caspase 6. Those are what? Executioner caspases. So let's start with this one. Caspase 9. So this apoptosome is going to activate what? Caspase what? Caspase 9. Very, very important thing. So remember, so make sure that you understand to say under mitochondrial pathway or intrinsic pathway, the caspase that's going to be activated is what? Caspase 9. Okay. Now caspase 9 is going to do something else. So it's going to activate other caspases as well. So this caspase 9 is what we call an initiator caspase. So an initiator caspase is going to activate the executioner caspases, like what? Caspase 9 and what? Oh, caspase 3 and caspase 6. Okay, so this caspase 9, what's going to do? It's going to what? Activate what? Caspase what? Caspase 3 and what? 6. It's going to activate caspase 3 and what? 6. Then starting from caspase 3 and 6, then you are beginning an execution what? Phase. So remember that if you talk about phases of apoptosis, there are two phases of apoptosis. There is what? Initiation phase and what? Execution what? Phase. So this phase starting from what? Mitochondria, everything happened to mitochondria, back clicking, forming pores like that, up to activation of what? Caspase. Then that is what we call an initiation what? Phase. Then execution of phase start with what? Activation of what? Caspase 3 and the caspase what? 6. Okay. Basically, remember that we are talking about intrinsic pathway or mitochondrial pathway. So now ask yourself to say, caspase 3 and caspase 6, what are they going to do? So they are going to do something very, very important. But I'll just talk about two main things that they are going to do. What do they do? So they go to the nucleus and they will make sure that nuclear fragmentation what occurs, okay, or nuclear breakdown. So if they go to the nucleus, they start activating enzymes inside the nucleus, which we call endonucleases. Okay, so what do they do? If the first thing that they do is, if they go to the nucleus, they activate what? Activate endonucleases like DNase, and what do they do? Then they cause what? Nuclear fragmentation. Nuclear fragmentation. Okay, so this is what they do. Nuclear fragmentation. Okay. So that's the first thing that they do. What else apart from that? So what else do they do? Okay, they do something else also. They are going to start like activating now, instead of endonucleases, they start activating proteases that will start breaking down what? The cytoskeleton of the cell. So they start activating what? Protease. Then what do they do? The protease, what do they do? They start breaking down what? Breaking down cytoskeleton. They start breaking down the what? Cytoskeleton. Now that's where the apoptosis end from for what? Intrinsic pathway. Okay. So now, since intrinsic pathway we are done, let's go to what? Extrinsic pathway. So intrinsic pathway basically it means that it is within the cytoplasm, it's within the cell. That's where apoptosis is starting and what is going to end from. Okay. Then if you talk about extrinsic basically starting from outside the cell, then the apoptosis, the initiation or the beginning of apoptosis starting from outside, they need to end from inside. Okay, that is the extrinsic pathway. So under extrinsic pathway, basically it's not that difficult as well. So if you look at extrinsic pathway, there are some receptors that are there. So there's this receptor, okay? This receptor, very important one. We call it a fast receptor. We call it a fast receptor, okay? This is what we call a fast receptor, very important. And fast receptors are under what we call tumor necrosis factor receptor type 1, okay? TNF receptor. So they are under a certain family of receptor called tumor necrosis factor receptor 1, okay? And a good example of that tumor necrosis factor receptor 1 is what? Fast receptor, okay? Or sometimes fast receptor is so sometimes called the death receptor. Why? Because it causes what? Apoptosis, okay? So now, this fast receptor, if you put, there is also another name for fast receptor, don't get confused. So it is also called CD95, okay? CD95 or fast receptors, that's one, just the same thing, okay? Now let's say there is what? A stimuli here. So here there is a stimuli here. Here there was a cause because there was something wrong with the mitochondria. Maybe there is mitochondrial damage, DNA damage, a lot of causes here, okay, causing that's stress to mitochondria. But here there has to be what? A stimuli here, okay? A stimuli here, there has to be a stimuli here that's going to stimulate them, okay? That stimuli, it can come from a lot of what cells. So I'll just pick one good example, which is maybe the T cells, okay? The T cells, if they are activated, let's say this cell is viral infected, okay? It has a virus inside, and the leukocyte, basically the macrophages, they are able, the T cells, they are able to what? Recognize that viral infected cell, okay? And those T cells, they have a ligand on the what? On their cell membrane, okay? So they express a ligand on their cell membrane, and what we call that ligand, we call it CD95 ligand, okay? Or fast ligand, okay? So what we're saying is that if this is a white blood cell, it will come here and express what? The fast ligand that will bind to the what? Fast receptor. So what we're saying is, this is what? The fast ligand. So this fast ligand, where is it coming from? It is coming from the what? Activated T cell. That has an intention to what? To kill the viral infected what? Cell. So that's where it starts from. So if this is what activated like that, then this fast ligand will bind to what? Fast receptor or CD95. Now this fast receptor, it has this part which we call the death domain, okay? So we call it death domain. So this part inside here, it has what? Death domain. This same death domain, it has what? The the adapter protein, okay? So this death domain, it has what? The death adapter. The adapter protein. So it has an adapter protein. Again, this same adapter protein that is combined with the death domain of the fast receptor, okay? It also has also the what? The adapter protein as well, or it has the what? The death domain as well, okay? Protein, okay? So this death domain of the what? Fast receptor, or the CD95, has its own adapter protein. But again, this adapter protein, it has what? Its own death domain. Don't get confused, okay? So this adapter protein, it has what? The death domain, its own death domain here, okay? And this death domain, we call it what? FAD. We call it what? FAD. What is FAD? Fast associated death what? Domain. Basically, it's a death domain, okay? Very, very important. Just to recap again, remember that this is a leukocyte that has a what? Fast ligand. This fast ligand is going to bind to the what? Fast receptor, or the CD95. This fast receptor of CD95, it has a what? Death domain inside there in the cytoplasm. Okay. This death domain combines what? Adapter protein. The adapter protein has what? As its own what? A death domain, which we call fast associated death domain. Okay, very important part. Now, this fast associated death domain, which is FAD, what does it do? It does a lot of things. Okay, it does an important thing, which is what? Activating the what? A certain caspase, which we call the pro-caspase what? 8. So this FAD, what is it going to do now? It's going to what? Activate caspase what? Caspase 8. Very, very important. And if you can see from this side, from the intrinsic pathway, there is caspase 9, activation of caspase 9. From extrinsic pathway, there is what? Activation of what? Caspase 8. Basically, you need to know the difference. So now remember that just from just like the extrinsic pathway, if you activate now a caspase or an initiator caspase, then that's where now initiation what ends from. So the initiation phase ends from what? Activation of caspase 8, but from the what? Extrinsic what? Pathway. Okay. So now, if this happens now, what's going to happen? Just the same thing now. Then the exclusional pathway or the exclusional phase, then activation of this. Remember that if you activate caspase 8, then caspase 8, what is it going to do? It will do the same thing as what? Caspase 9. It will start activating caspase 3 and 6. Then caspase 3, caspase 3 and 6, what is it going to do? To start activating what? Endonuclease and what? Protease. Endonucleases, like what? DNS, what do? They will initiate nuclear fragmentation. Then what about protease? What do? They will start breaking down the what? Cytoskeleton, leading to what? The death of the what? Of the cell. Okay? So that's where the mechanism of apoptosis ends from. Now, there are some other things that you need to understand as well. Okay? So there are also some other things that you need to understand. Okay, under apoptosis. The question is, you're going to write apoptosis as Z occurred now. So how do those apoptotic bodies removed from the what? From the body, or from where apoptosis is happening, okay? Because this has to be removed immediately, because if it is not removed, inflammatory response is going to occur. But under apoptosis, remember that we said there is no inflammatory response. Basically, so the cleaning up of these apoptotic figures, because after apoptosis, everything will be turned into what? Apoptotic bodies, okay? Or apoptotic figures. So now, how do you remove apoptotic bodies or apoptotic figures? So here is the thing that we'll talk about here. So removal of apoptotic bodies or figures. So how do you remove apoptotic bodies? Basically, they are removed by the what? White blood cells, but specifically we're talking about macrophages. So macrophages are the one that play an important role in the removing of what? Apoptotic what? Bodies. Okay. Now the question is, how do they differentiate from a cell that is just normal, that has not undergone apoptosis, and the one that has undergone what? Apoptosis. There are some various mechanisms that happen so that the white blood cell or the macrophage can recognize that this is a cell that has undergone what? Apoptosis. Okay. Two important things. Okay, the first one is what? A cell, an apoptotic cell, a cell that has undergone apoptosis. They turn something. Okay, they on the cell membrane. Okay, on the cell membrane, they put a certain signal. Okay, a very important signal. So what is that signal? So it's called phosphatidyl serine. Okay, so it expresses something like phosphatidylserine. So basically, this phosphatidylserine, basically in the normal cell that hasn't undergone what apoptosis, they are expressed inside inside the cell, basically in the inner membrane of the what, of the cell membrane. Okay, but a cell that has undergone apoptosis, this phosphatidylserine is going to be expressed from what, from outside. Okay, then if it is expressed from outside, so that is a signal showing that okay, this cell has undergone apoptosis. Then white blood cells will come there and recognize those phosphatidylserine and initiate what, phagocytosis. Okay, so phagocytosis, basically they are going to what, start eating up those apoptotic bodies. Okay, what else? Okay, so they also secrete something. Okay, so they also secrete soluble substances. So those soluble substances, they go to what, white blood cells or macrophages, basically. Sometimes also neutrophils, they can be there. Okay, so if they secrete those soluble substances, they're going to recruit what, white blood cells from what, a distant site or maybe from nearby site, so that they can come where apoptosis is happening, so that they can be cleared out. Okay, and what we call those substances, thrombospondin. Okay, thrombospondin. So thrombospondin, these are just soluble substances that are going to recruit what, white blood cells, so that the apoptotic bodies can be what, clear out. And maybe sometimes there are also some other, maybe other things that are also important, like antibodies, complement system. They also help in the clearing out of what, the apoptosis. That's why you see that under apoptosis you can't see what, an inflammatory response, because this clearing process happens immediately after apoptosis. A very what, fast process, because it is preventing the what, the leaking out of what, these substances that are within the cytosol of the cell that has undergone apoptosis. If they leak out now, they will start causing an inflammatory reaction. No wonder this clearing out of what, apoptotic bodies is very crucial in the apoptosis what, process. Okay, very important. Now from this point, since you are done with apoptosis, now make sure that you stay tuned, so that in the next video, now we'll talk about the mechanism of what, cellular injury. So those causes of cellular injury that we talked about, we'll talk about under molecular level now, the mechanism like the ATP, okay, and the other things, as well as we will talk about the other clinical relevant of what, cell injury, like perfusion, cell injury, those types of what, things, those special things. Okay, now since we are done with apoptosis, make sure that you subscribe so that you can be updated with upcoming videos.